Chapter 2, Section 12, the other forms of taxes. Section 11 focused, talked about taxes, the run and tumbles and what, or the just primarily run if it's a polar flagellated bacteria. But that focused mostly on chemotaxis. Keep in mind, there are other forms of taxis. Osmotaxis. How much water is there? Is this going to be, isotonic, hypertonic, hypotonic, aqueous solution? It's going to want to move, so there's, how much water is there? What's in the water? Is this going to affect water moving in, water moving out of the cell? How much oxygen is there? How much light is in a given area? The phototaxis one, we can sit there, and that one's really easy to study because you can look at microbes and take samples as you expose certain areas on a liquid broth to light. Here's a high intensity light in one area, low intensity light in a different area. We've studied movement of the microbes as they move across an agar towards light, given time. How far do they move? So this is pictures that have been cut showing, within a few minutes, almost an hour, hour and a half, 2 hours, how far the bacteria have moved across the surface. We've also studied which wavelengths show us the most chemoattractive abilities. We've done it with oxygen, and like I said before in the last section, we've also done it with magnetic fields. The bacteria will align themselves within the liquid broth or on an agar plate surface based on what the magnetic fields are. This is not all of them. These are the only ones that, contain those iron granules inside their cells.